Okay, thank you for coming to what is now the fourth research ethics workshop that we've done at Langara in about 4 years. These presentations are published on our YouTube channel. There'll be an opportunity for you to ask questions. Today's presenter is Patricia Birch. She's from UBC. She's a clinical associate professor in the Department of Medical Genetics. Genetics at UBC, is also a researcher and manager at Children's and Women's Hospital. So I'll let her fill in details about herself and we'll carry on now. Thanks, Patricia. Thanks for being here.
Thank you very much for inviting me. I think probably the most important point is that I'm actually a nurse, and much of my life has been spent surrounding issues of advocacy and justice and equity, and that's a personal passion. Like, probably that's the most important thing to know about me from where I'm coming. I should also say that I think you advertised that Jenny Morgan, who is the Gitsan co-lead on the project that I'll be alluding to on and off in this presentation, she was going to be hopefully providing a lot of input to the discussion, but she, the last minute, couldn't make it, so you're stuck with me. But that being said, we do need to absolutely acknowledge and keep in mind whose territory we're on, and acknowledge the Coast Salish First Nations. And I think you have a particularly strong association with Musqueam in Langara, if I'm right? Yeah? Start off with a little bit of an outline, so we can see where we're going to be going. I'm going to be hopefully talking a little bit about why you want to do genomic testing and how it's done and the limitations, because the limitations have a lot to do with the ethical implications. I also want to talk about something that's maybe not so much in the literature, and that is the difference and that gray zone between research genomic testing and clinical genomics testing, because it is very gray. Much of what we do is in that zone. So that has real ethical connotations and implications. Then I thought we'd look in a little bit more depth at some of the ethical issues and challenges that affect everybody who's being tested with genomic testing, and then move more specifically to how genetics have influenced Indigenous peoples, including history, which many of you know probably quite a bit about, and then some specific research examples with my project in UBC, REB, which, sorry, they're not bad, those of you who are there. And then I'd like to take things a little bit beyond the famous Chapter 9 of the Tri-Council Policy Statement and look at other frameworks that we could be looking at, and specifically cultural safety is one that Jenny feels very strongly about and has taught me a great deal about. And if we have time, I'd like to spend some time showing you a little bit of various bits of videos of the work that we've done. But most important, I would really like this to be interactive. I mean, I'm not an expert in either Indigenous health or in ethics, or in genomics for that matter. So let's make that clear. So there are lots of experts, thank you, Jennifer, in the room who will help me with all of these aspects. So, but jump in if I'm saying something that makes no sense. Just please contribute to the discussion. That would be great. This is apparently supposed to be a workshop right now. Yeah. Okay. Genomic sequencing. I'm sure that you've all heard about it. If you haven't heard about it in the press, I don't know what planet you're on. But it is indeed extremely powerful. It's also relatively new, and it used to be extraordinarily expensive, but because the cost is rapidly diminishing, it's now being used a lot. Why do we want to use it? Well, first of all, I should say that it's a single test that's sort of one size fits all. It looks at all the genetic information at once. If we're talking whole genome testing, we're talking about the whole genome, all our genetic material. If we're talking about whole exome sequencing, which you may also have heard of, that's just 1% of the genetic material that codes for our 20 or so thousand genes. So beyond that 1%, in the rest of the 99%, we've got controlling regions, we've got a lot of regions that we actually don't understand. So it's a very new technology, and we are really learning, with what we're seeing, what we're able to do right now with it is really tip of the iceberg stuff, so that's important. But already, we have a high diagnostic rate. So there's a project that I'm doing at the moment in Dr. Jan Friedman's lab, and we're sequencing 100 trios, trio being an affected child and parents, and 2 normal parents, where the child has atypical cerebral palsy. And these are children who've had basically every test in the book. Many of them, they have charts that are this thick. And we're picking up a diagnostic rate of, about 50%. So why do we want to diagnose this? Well, obviously, ideally, we would like to find a treatment. That's probably not very realistic for the majority of people. Only a very small percentage of people will be in treatment for. Mostly biochemical things, where you might think you can maybe replace something that might be missing. But sometimes, more often than that, a diagnosis might lead to changes in management. And you can think of everything from, okay, well, is there a better drug for this? It's not going to cure the child, but maybe this drug would be better, to, we know that this child will ultimately become blind. Now we have this diagnosis, for example. So that has implications for the care you give to the child and to the family now. So that's management. And we can't underestimate the value it has for the family. So there's so many families who have blamed themselves. Mothers tend to do that. It was something I did in my pregnancy, and you can relieve that. Or you can provide information that will help other family members to say, okay, well, I know how to interact with this child now. We know it's the matter. Well, we'll just accept the child, and now we can just get on with caring for the child. So there are lots of profound values that I'm sure you can think of many examples for. So how does genomic sequencing work? Yeah, go ahead.
Just a question, and if we go back to the first slide there.
The last one?
Yeah, the first one.
The very first one?
No, the one you just were on. That one, yeah.
Yes.
Would you describe this as a screening procedure? How does this differ from screening, or does it?
No, I wouldn't. It's certainly not used as a screening procedure right now. So right now, it's expensive, and what we typically use it for is when there's a high degree of suspicion that there is a genetic cause to the child's or the person's, the adult's, child's, anybody's condition, and that we've run the gamut of cheaper and obvious tests, and now we basically have, this is sort of approaching ultimate to what we have right now in terms of genetic testing. Is that?
Okay, thanks.
So, yeah, I mean, that's a good question, and screening test is something where we may be going, thinking
That's what worries.
And we can absolutely talk about that later, because I mean, we know about the screening, for example, that every newborn child gets, and that's based on evidence that there's a certain proportion of children with this that we can pick up and that we can treat with this, right? So there's a lot of evidence that is required before you implement something as a screening test, and we're very long way from that, but that's a good thought to keep in mind. Thanks. Okay, so sequencing. Probably to put the workshop in context, we need to have some understanding of how sequencing works and its limitations, because that informs the ethical issues. So we take a blood or some other tissue, we send it away, and magically it comes back sequenced. What they do is they sequence little bits of it, and then they join it together computationally. So we have this sequence that represents our body's instructions for functioning, and it's 3 billion DNA pairs. Now, sometimes just one single letter change can have quite devastating effects on somebody. But there are a lot, and that's a simplistic sort of view where, you know, there's an A where there should be a G. But there are a lot of other alterations that we look for. Deletions, insertions, really complex DNA rearrangements that we're only really just beginning to understand some of. And you've probably heard of repetitive elements, or if you have Huntington's disease, that's a trinucleotide repeat, so 3 little letters repeated, 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 repeated. So that's one we know of. But I was talking to the postdoc in our lab the other day, and also she's looking at hexanucleotide repeats. Well, that's what I mean by we're really just scraping the surface of what we can do with whole genome sequencing right now. So the other important point to note is that the human genome is really variable. So we're only 99% the same as each other. So you and I differ probably by 3 million, 0.1% if I've done the math right, bits of DNA. And most of us, probably with my possible exception, are relatively normal in this room. So we make the assumption that that is normal variability. It's just, we expect to see it, and it's normal. What makes me, you know, blonde and blue-eyed, and somebody else have black hair and green eyes, or whatever. You know, those are the differences that make us human. It's the richness of human beings. So, for example, if we were to sequence a little bit of my DNA, we might see an A here, and maybe John's DNA, we might see a T here. So, how do we know if these are both normal variants, or if this could be getting me into trouble, or John into trouble sometime down the road? So that's the important issue. How do we determine if any one of these changes is just normal variability, or if it could be pathogenic? So we need to have a reference to compare to. So, I mean, if I'm diabetic and I prick my finger and I test it on my meter and it says 3, then I know I'm low, or it says 8, I know I'm high. Well, the reason we know that is because lots of people have had their blood glucose tested, and as a consequence of that, we know what the mean is, what the standard deviation is, and so forth. So when you go to Life Labs and they do your cholesterol and they say, ooh, you're outside the normal limits, you know that that means that you're beyond 2 standard deviations, because that's how they define it, of the normal, of the mean. But unfortunately, it doesn't work quite that well with, in fact, it doesn't work at all, to use that approach with whole genome sequencing, because there isn't a, there isn't a single perfect human that we can use as a reference.
There's no gold standard?
There is no gold standard, exactly, yes. Despite what any of us may think in this room, we're not a perfect human being. Yeah.
Shattered dreams.
Shattered dreams, I tell you, yeah. Sorry about that. So what we do instead is we look at a database that has thousands of healthy people's sequences in it. And then we might see that A is something that is in 25% of people, and the T that we referred to earlier was in 75% of people maybe. So if it's that common, then it's obviously a normal variant. So we have to compare each patient's millions of variants to those seen in a reference database. So we should maybe step back a minute and ask, where does the variability come from? So basically, there are kind of 2 choices. I'm sure Jennifer can come up with some others, but you know, in simplistic terms, either it's a new variant in the person being tested, which probably means it was something that happened in the egg or the sperm or early on in embryogenesis, or it's something you got from mom or dad. And of course, if it's something that you got from mom or dad, maybe it was a new variant in mom or dad, or maybe they in turn got it from their parents or their parents, and so on and so forth. So the sort of corollary to that is that we are most similar to the people we're most closely related to. So we're most similar to our parents, our siblings, kids. We're pretty similar even to our distant cousins. And we're relatively similar to people of our same ancestral group. But if you looked at me and you compared me to people of Northern European origin, I would be much more similar to them. You know, within this 0.1% we're talking about, right? So it's all relative. Than to, say, somebody from Asia or Africa or Australia, Aboriginals from Australia, or indeed Indigenous people from Canada. So, therefore, keeping that in mind, the crucial thing is that we need a reference database that contains many sequences that are close to the ancestry, as close as possible to the ancestral group as a person being tested. Have I lost anyone? That makes common sense. If you're writing furiously, I'm really happy to give you my slides afterwards.
There's a connection between the heat and the rain.
It is true, actually. It helps you to sort of, yeah, keeps you awake too, right? So the international database that is probably most commonly used is something called gnomAD. And gnomAD is a coalition, a resource that's spread out of, I guess, the Broad Institute in Boston. And it's really a collaboration of a load of international investigators. And from their website, the purpose is for the benefit of the wider medical community without restriction on use, which is great, right? And right now it contains about 150,000 anonymized genomic sequences. So that's also great. Lots of sequences in this reference database to compare to. And again, from their website. Yeah, John.
So I have a question. So, I mean, is this something, it seems to me this is the need to acquire data from ancestral groups, right? That's something that's dynamic, isn't it, right? In the sense in which over time, you know, given flows of population and so on, will you have to be constantly updating your sequencing, given that you have people, you know, marrying across ancestral groups or reproducing across, better word, right? Across that. So I'm just curious, how static is the need for sequencing within particular ancestral groups, and how reliable is it or useful is it given population flows?
Yeah, okay, I think I see what you're saying. So in our lifetime, we're not sort of changing sufficiently quickly that anything that we enter here will be relevant. But don't forget, it's not where you live now, but where your great-great-greats came from. And if your great-great-greats came from a bunch of different places, then that's good because we have a bunch of different places in this database. So that's a really important point. Thank you. Yeah. And these are some of the examples of countries that have contributed.
Can I just ask something then? How, and I take your point that doesn't make a lot of sense, and I'm just speaking as someone with parents from completely different countries.
Yeah.
So our ancestral groups would probably just be quite divergent. So then how would we define an ancestral group?
The human race is probably the ultimate measure of ancestral group. But I could take my children, for example. They were born in Canada. 30 years ago today, I was in labor. That's just beside an hour away from delivery. Sorry. I just realized that. Sorry. So they were born in Canada. I'm obviously from the UK. Their father may be from somewhere totally different. Their father may be Asian. He's not, but he could be from South Asia. And we can either know that, in which case that's information that's helpful, or we can trust that there are South Asian genomes in here. And that kind of brings us to the next point. And then ask me if you don't get it after the next slide. If we take the countries and the distribution in gnomAD, what we see is something quite probably not unexpected if we think honestly about where research is done. These are people of European ancestry. So the vast majority of gnomAD is full of people from European ancestry. There used to be very few people from African ancestry or Asian ancestry, the yellow and gold down here. Now those are increasing in number, so it's becoming more representative of the world's population. The problem comes in that 2% at the top there. So that 2% is other. So if you wanted to find Maori, if you wanted to find Australian Aborigines, if you wanted to find Maya, if you wanted to look at Canadian Indigenous peoples, that would be where you would find them. And as far as we know, to the best of our knowledge, there aren't any Canadian Indigenous people in gnomAD. So we go back to that thought, that big screen we had earlier, that you need is a reference database, something that's as close to your ancestry as possible. This is where the problem lies. So there are no First Nations, Inuit, or Métis, or virtually none in the reference databases. gnomAD isn't the only one, but it's the big one. So that means we have significant challenges in interpreting some of those DNA variants.
I have a question about the previous slide. That's the distribution. Are you showing them? Explain again what this bar represents.
Right.
Is this in the 150,000?
Yeah, so thank you, thanks,
The
yes.
blue would represent the proportion
Yeah, so whatever it
that
is.
were
70%.
European ancestry. Is that
Yes,
correct?
that's right,
And
yeah.
could you just describe the one, the Ashkenazi Jewish?
Why is that so big?
Yeah.
I can hypothesize, I don't know, but I can hypothesize. There are probably a couple of reasons. First of all, there are some very specific conditions that are quite common, or are much more common in people of Ashkenazi Jewish ancestry. And Ashkenazi Jewish population has been extremely strong in advocating for themselves. And so my guess is that they have actively worked for this. So it's a guess,
Okay.
but it's probably an educated guess.
If I may, Ancestry.com, right, or a similar,
23.
there was
Yeah.
in National Geographic, one of my cousins sent in a sample, right, to have his sequencing done.
Yeah.
And they sent out a report back to us. And in the report, the first report they had that we were in that Ashkenazi Jewish, right, we were in that group. Is that like 100%?
No, it's not even close.
Right. Can they do that with a swab from your mouth? Can they really tell you you're in that group? No. Yes or
They
no?
are getting better at it. I'd say yes and no. So what it means is that you share some variants. Don't forget, 23andMe is not a whole genome sequence. It's looking at various spots, or Ancestry.com, I presume works the same way. I haven't actually looked, but yeah. So it's looking at little spots throughout the genome, looking at little kind of indicators. And so maybe there's an indicator there that whatever your ancestry actually is, it's shared in common.
Yeah, actually, on that too, as a Brazilian with 100% European background,
Yes.
let me ask you, what is Latino?
That is a
Because I think that
that's a self-identified U.S. political thing. So I don't want to go there because when I am involved in U.S. research, I have this really obnoxious form that I have to fill out, and it says, "Are people white?" Are they African? Are they Hispanic? They're part of the human race, so I usually refuse to fill that out. But I don't know how they define Latino. It's a good question.
No, because I would suspect that that 2% would increase quite a bit if you looked at it Latino and realized that what is Latino is pretty much the ancestry would be indigenous people.
Could well be, but it's probably not Indigenous Canadians, right?
Absolutely.
Yeah,
I'm just kind of looking at Latino specifically and
yeah.
saying, oh, interesting.
Yeah, that's a really good point, and that's I don't know the answer to that one. So yeah, good question. Yeah, thank you. Out to So here's the synopsis of all the previous slides, that either clinical genomic testing or research genomic testing of Indigenous Canadians is compromised. And then 1 in 20 of us. So that's not good enough, is it? I'd like to give you a real life example that illustrates this. And this is the family that actually, when I heard about this case, got me started on the research project with Jenny Morgan to talk with Indigenous people about their perceptions and values of this, around this issue of inequity. So this is a family, I've obscured the details, just obvious for ethics reasons, seem like a good idea with this audience. This is a First Nations family from a rural reserve, and they came down to Children's Hospital. Their kids had some sort of a metabolic-ish problem. Lots of testing done, not in medical genetics, in another department, couldn't find out what's the matter with the kids. Ultimately, they turned to exome sequencing. And when they got the results back, they found that both the children had a variant in the same place, in the same gene. And it's a gene that we know very well because it's associated with a very serious health condition. And we know that that gene had nothing to do with the condition that the children presented with. So then we say to ourselves, okay, so what is this? So you throw it at the usual databases, and you don't find that variant in either databases of healthy children, like gnomAD, or databases of people with various illnesses. So what is it? We know that other variants in this serious gene will create a lethal problem. So, is this an incidental finding? Meaning, is this something that we weren't looking for, but we found it, and it's real, and we need to tell the family about it? Or is it just something of unknown significance? So, the individuals who saw this family told the family about it. So the family came into Children's Hospital with one problem, and they go out of Children's Hospital thinking that they have two problems. And not only that, but that the second problem is a really bad one. A couple of years later, they came back, and this time they came to medical genetics. And meanwhile, science has moved forward, and geneticists deal with this more often than the original folks maybe who saw this family. But I think mostly it's just that our understanding has improved in that period. And the geneticists said, you know, your kids are perfectly normal with regard to this condition. It would have shown up by now if it was going to. I suspect that this is just something this variant is something that's normal in your ancestral group, and we just don't have the data to tell you that. So I'm going to be fairly confident that this is the case, although I can't be 100% sure. So that's now what the families have gone with. So now they have some sort of belief that the geneticist is being reassuring, but it's still a variant of unknown significance. So that's what this family is actually what got me started on this research. So, okay. Do you have any thoughts, by the way, about the fact that this family was told about this in the first place? What do you guys think? There's no right answer, I'm sure, but
I guess if there's nothing that can be done, the question is why tell them? I guess. I don't know.
Actually, this came up. I taught the graduate students in the medical genetics program at UBC, and this ended up being one of the issues that the students were grappling with. And a couple of them wrote some really good research papers suggesting that if you're going to respect the autonomy of the people who come for this kind of testing, then it's sort of the duty to at least inform them and then to give the information that you suggested, they sent them home with saying, look, we don't know what this means, but that to withhold it from them might actually engender mistrust between the patient and the geneticist, or the patients and the physician. So the class collectively thought, well, probably reveal the information with a proviso that, we don't know what it means.
yeah. I think I'm really glad to hear that. Was it genetic counseling students?
Yeah,
Yeah.
counselors.
I'm really glad to hear this because I think that's where the majority of patients, your average everyday people on the street are going. To quote Isabel Jordan, who's, you may know, she's a patient advocate and chair of the Rare Disease Foundation. She says, treat us as adults, please. Give us the choice. But it's difficult, and I think you'll find that from the clinical geneticist perspectives, they're going to be more conservative. And of course, again, this is not a binary scenario. It depends on the strength of the evidence, right? And people are going to make different decisions under different situations, and these are decisions that they make every single day. So, yeah, it's a really tough one. And that's why genetic counselors exist, to try to support people with these decisions and make sure that they're updated with the information and do what they can to make sure that they're informed. So, maybe I'd just like to ask you, does anyone know how you would get your genome sequenced right now? What would you have to do if you kind of thought that there was something better? Anyone have a clue? Yeah. Do you think it's like your cholesterol test, probably? Can your doctor just order it?
Private clinics?
That is definitely one solution. So yeah, to not hold you in suspense, you can, your average GP or pediatrician can't just say, okay, I think he needs a whole genome, or I think he needs an exome, let's test this. No. So medical geneticists spend a lot of their day, a lot of their time, writing elaborate letters to MSP and to try to persuade them that this particular family deserves, needs, can benefit from sequencing. Under no circumstances really do they consider the value to the parent of just knowing. That's not considered by the people that make those decisions. It's really a very clinical decision. Sometimes it seems like flipping a coin, but anyway, we won't go there.
So is that a request for special funding for, in particular cases?
Yeah, all of them go that way.
All of them go that way,
Go that way.
and so what, can you just say a little bit about what the decision-making process is to allocate resources then on the list?
No, we can't.
But do resources get allocated?
Yeah, I think so. But I should also say that this was last month's model, and there is a new model that's being developed right now. So that part of it's going to get different and probably better. Because if you have, imagine if you have a geneticist and a genetic counselor and maybe a cytogeneticist involved in the decision making, then you can probably get a more informed decision. That's probably where we're going.
What are the costs? What does it cost now to do this?
Oh, it depends on what test you're ordering and how many people you do. So, I mean, if you do one person in the family or a trio, which is whether you do exome or whole genome or a panel, but anywhere from 1,000 to 10,000. So we're not talking about peanuts here. It's not your $40 yen.
If tests are done through the MSP, is it done through in publicly controlled laboratories? Or
Yeah, they're
or is it
sent
third-party
to
private labs?
clinical laboratories, typically in the US or Europe. They're getting here. There are alternatives here, but the majority right now go to the US and a few go to Europe. Yeah, and they're clinical labs, so you get a clinical report back. Yeah. So you pointed out, I think, yeah, the alternative is private pay, and some people, when they get that letter back and it's refused, which is a lot of the time, then some families do opt to pay privately. And then there's the cheap option for everybody, which is research. So there were a lot of research programs. We just finished something called Causes. The Causes Clinic sequenced 80% were exomes and about 20% were whole genomes, 500 trios. And so if you can get your child into a research protocol, then not only is the family really happy about that, but also the clinician is happy. Saves you writing a letter, and you have a better chance of actually getting something. So that's how you get sequenced right now. You should see the very creative ways that people try to fit their child into a, yeah.
So if you're in a research study, how does that relate to their clinical care?
I'm so glad you asked that. That's the next couple of slides. No, that's great. That's a superb question. Let me just reveal all these. So that fits right into the example of day-to-day ethical issues. So Imagine is the study that I mentioned earlier, the name of the study. We have to have these names because otherwise we can't remember the titles that on the consent forms that are this long. So Imagine is the atypical cerebral palsy study where we're getting about close to 50% diagnostic rate. So this is how it works. We do the sequencing on a research basis. We find something. That's a research finding. That finding can't go into the clinical chart. So now we have pressure from the clinicians and of course from the families, rightly so themselves, to give me a clinical report. Give me a clinical report. What does this mean? Something that I can act on. So we take this finding and we send a new sample that meets the clinical standards to Life Labs Genomics in Toronto, and we say, hey, Life Labs, look here and tell us what you see. We want you to look for this. So they look for that, and then a couple of things can happen. They can say, yeah, we see that too. Here's a clinical report. It's a slam dunk. It's simple, straightforward case. Fantastic. Quite often, though, what happens is this. When we decide that this particular thing is the issue in the child, it's not just looking at that. We're comparing this particular variant to the child's phenotype, to their signs and symptoms. Does it make sense? Is it in a genetic pathway that we would expect to see this phenotype? That's obviously the first step. And then there are mechanisms that you can sort of upload questions. Has anyone seen this particular thing? You put it into the stratosphere and you get a response back from someone in Germany, and they say, with luck, yeah, we've got 4 families who have the same thing and it looks exactly the same as your child. And then you go, great. And that really firms up your research finding. But then Life Labs does that. Does look, and they say, yeah, we see the thing. But we had to call it a variant of unknown significance. That is because that association between the genotype and the phenotype isn't solid enough in their stricter clinical lab minds to be able to call it a clinical for sure. So that leaves another gray zone for people to be in. And so, I mean, the other thing is, if you think about it, we're now, this research study, which was funded by us, we taxpayers, CIHR, and Children's Hospital Foundation, various others, is now not only paying research dollars for a research report, but we're also paying for a clinical report. So there are interesting ethics around that too. That's sort of it's a bigger level than I can think, but it does cross my mind that we could be sequencing a whole bunch more people if we didn't have to go to Life Labs in Toronto to confirm these findings. But we do, and for good reasons.
But you're sick. Sorry, go ahead.
Yeah, go ahead.
I was actually going to go back to something else, so if you want to continue with this.
Yeah, no, I actually lost a little bit. I was following you up when you said that, so you validate the results with Life Labs in Toronto, but you're saying that that's not a helpful clinical thing because you are recognizing something that might be actionable in Life Labs and saying no, or?
That happens, yes, and
then what?
So that's the then what. So the then what is, do you sit down with the parents and you say, we're pretty sure that this is what it is, but I think that's typically the honest and open approach, including the parents in the discussion. So it looks like this is what it is, and we can't get clinical confirmation now, but maybe in a year from now we will be able to. So things which today are variants of unknown significance, hopefully in a year or two, we will know that either they are indeed pathogenic or they're benign. And likewise, we are reanalyzing some of the data from that causes project, which I said had 500 trios. And we are reanalyzing them with more reference data and different techniques. And now we're finding a small percentage of diagnoses that we can make that we couldn't make earlier. So it's a moving target as well. So as if it's not bad enough that it's gray, it's also moving gray. It's about 50 shades of, I don't know, ultramarine gray, right?
Yeah. How does the CREB deal with this right now? Like, don't ethics boards usually require that validation? And then, but now I find it fascinating that you can have this, maybe really clinical results that aren't validated in a clinical database.
We could.
That, yeah.
Sorry, go ahead.
Yeah, no, I'm just seeing that we're not ethically solving anything by requiring that. If in fact it can still be that there is an actionable item.
Yeah, exactly. And I think the main thing is really patience. And maybe I'm not the most patient person in the world, but it frustrates me to see that. But I also know that it will get better because our research findings will be published, and then ultimately there'll be 10 kids with this, and then it will be in the Clinome, and then oh good, then now we can rubber stamp it as being a clinical finding. But it takes time. Yeah.
There's a gray zone with respect to what counts as a clinical finding too, and I think the fact you're talking about, right?
Yeah.
So it's one thing for the researchers to have a strong hunch or to believe that they found something. Researchers do that all the time.
Yeah.
And the broader scientific community says, well, let's wait a little bit, right?
Yes.
So that's not necessarily unreasonable.
No, it's not, and it's a conservative approach to research. Yeah, we could take another extreme. We could have a research project where, we're sequencing 100 trios for children who have atypical cerebral palsy. We're not getting any results back though. It's just a research study. Now, I can't imagine many people would actually be too keen on that. And that was one of the earlier approaches to some of the earlier methods. And, parents wouldn't stand for it, and why should they? For the reasons that we talked about. In this era where it's really an era of participant-involved research, we need to go this route. It's just that it does create difficulties. So, and this is for everybody, it's not just for First Nations. I also just want to talk briefly about what I call one-offs, because there are a lot of them. Rare diseases are rare, but if you put them all together, they're very common. So a child comes in and sees one of the geneticists in medical genetics, and the geneticist writes his letter thinking that this kid needs a benefit from a sequence. Miraculously, it gets funded, he does a sequence, and he gets something back. It's not something that he's seen before, remember it's a rare disease, it may not be something that is published in the clinical literature. So he might, or she might, go searching through colleagues throughout the world. And you might find someone in Holland who says, you know, I had somebody who had that, and I tested the sibling, and I found this. You might want to think about that. Okay, so now our geneticist goes, oh, that's really cool. Calls back the family, tells them, says, hey, we should probably test the sibling. So now is this clinical care or is it research, right? So the further remote you get from this, now when you start making mouse models or fish models of the same thing, clearly it's research. But there's no geneticist who has the time in the world to write a research ethics board application for every single patient like that they see. So what happens instead is we have, about 10 years ago, we invented something that probably research ethics board was nervous about. You've heard of the GUARD study? Maybe. So GUARD is a sort of an umbrella protocol and consent that has 30 or so co-investigators listed on it. These co-investigators are all clinicians. And between them in the last 10 years, they have enrolled 300 of these one-off kids with rare diseases. So it's a sort of a workaround. I'm being perfectly frank to you. It's, you know, it works really well. It's the way that we can get care for these kids. But it, you know, it's also in that slightly gray zone, right? When does something drop out of being clinical care and now become research? So these are questions that there's no answer to, really. I don't think, anyway. But
Can I just ask a quick question? It seems like there is some mechanism of, at least for better or worse, regulating it from the perspective of clinics and research. But what are you calling private? Like, is 23andMe or molecular DNA private? And in that circumstance, who's responsible for interpreting that data? They just send you the data and expect
yeah,
you
that's a very good question. I mean, if somebody on the street sort of is interested about their ancestry and they go to 23andMe and they say, hey, medical genetics, can you explain why I'm Porty-Slavovian? And you're going to say, I'm sorry, that's not my job description. But on the other hand, if you are working with a family and you've tried to get MSP to fund this and they've said no, and then you present the family the option, you say, you know, it's really expensive, but it is an option if you want, then of course you're going to help them to interpret those results.
Does that mean that industry is also CLIA certified? Like certified?
Oh, yeah, I mean, we would send it to CLIA certified. I mean, we'd recommend CLIA certified labs. So the US is full of them. Yeah. Yeah, no, you don't send them to some
And to continue on that thought process though, so if somebody does have results out of a single type 1 organisms from 23andMe or Ancestry.com,
yeah.
does the clinician or the clinical genomicist say, we can't look at this table?
Again, another sort of grayish area. If you come and you say, probably depends on the severity, probably what you'd have to do is go to your GP and whoever and talk it through and then get a referral. But most likely, you already know that you have a family history. Let's say 23andMe, the Ashkenazi SNPs say that you're at high risk of breast cancer. Unless you're adopted, most likely you have a family history anyway. So, you know, maybe we're making more of a mountain out of a molehill here. The geneticists don't like doing that. They really like to sort of work it out properly in cold light of dawn, rather than having things thrown at them. They will typically not deal with those.
Yeah, just wanted to follow up on the one-off example. The sibling, for instance, like right off the bat, there's sort of, you know, mild sort of pressure to participate in this sort of research because that's one avenue to get that sort of information. Suppose that the recommendation is to get a sibling now. Now that sibling is already under sort of the normal pressure. Now the second set of pressures is, well, you know, if you don't do it, we won't find out what little Johnny has or whatever it is. So I wonder, it sort of ratchets up the coercive nature of participating, and then whether, you know, I guess those safeguards have to be built into the application of the informed consent and the ethics aspect of it.
I think REB does a pretty good job of trying to build those safeguards in. Of course, there are assets and adolescent assets, and the idea is that assuming that the individual is competent, then this should be a discussion that is independent. We know it's not. You're absolutely right. And then if the sibling is not competent, then yeah. Of course, you could also say that it will ultimately benefit the sibling, but it's talked about the numbers about that one. Yeah, it's tricky. Okay. How are we doing for time? I think I have to speed up here a bit. They're great questions, though. I thought we'd actually get back to Indigenous peoples. And this is a quote from Maori researcher Linda 3Y Smith, who's written an awesome book on decolonizing methodologies in research in Indigenous people. Word research is probably one of the dirtiest words in the Indigenous world's vocabulary. It's there's a silence, conscious of bad memories, raises a smile, that's knowing and distrustful. But if you think research is bad, then you should try being UBC genetics research, which is my choice. Are all of you familiar with the Rick Ward case? Or it's something I talk about a lot, but it's something that bears repeating because we really need to keep it, I think, top of mind. So forgive me if it's...
A quick go-through would be good for the video.
Okay, just real quick then. Yeah, so Rick Ward was a geneticist at UBC in the 80s. He became aware that the New Chalmers people have a high incidence of rheumatoid arthritis. And he went and he collected a bunch of blood samples, he interviewed a whole ton of people. He told them that he'd give the results back in a year, and he found nothing, and he never got back to them. Meanwhile, he published on various things, including genetic anthropology. He moved to Utah, and then as a result of his publications, he got a plum job at Oxford, still using and sharing the samples, obviously with no consent. Maybe fortunately for him, I shouldn't say this, because he died in 2003. I think he had something like 200 publications that were somehow related to this. And someone from the New Chalmers, people sorted this out, found this, made the link, and you can only imagine what happened as a result of that. The samples were ultimately returned from Oxford in 2004, and I recall boxes and boxes of data sitting in our department head's office around the same time. Larry Baird, to show you his reaction to this egregious breach of trust. Ward taught us to trust researchers like residential school, taught us not to trust the church. He uses like cheap guinea pigs. There were some upsides to this, if you can consider there to be upsides. This is the, I think, December 2004 documentation of the return of bloods. And the result of this, the New Chalmers Tribal Council and most other Indigenous groups at various levels now have research ethics attorneys. So there are many other examples. I'll just skip through. The Navajo banned genetic research for nearly 15 years, and then they reconsidered it when they realized that their new oncology center could use personalized genomics. But they did it under their terms. Any research was going to have Navajo oversight. They would decide on the source of research and who had access to the data, and multiple controls. So the question is, could something similar happen today? Here? What do you think? So now we have local Indigenous ethics boards. we have safeguards at the national institutional level. I think we also have hugely increased awareness and sophistication amongst Indigenous peoples. I think it still could happen.
I think it's extremely unlikely.
I think it's extremely unlikely.
Universities sign memorandum of agreement to uphold certain ethical standards.
Yep.
They have research ethics boards who are on top of these sorts of issues.
Yes.
They have duties to consult and so on.
I agree.
Consultation almost involves recognizing a property right with respect to the samples and sometimes the data.
Yeah. Yeah. Yes, exactly. There still could be. It could, but I think that the strongest part is actually that's the Indigenous awareness and sophistication. So anyone who's tried to do any Indigenous research will know how extraordinarily difficult it is. Yeah. You probably are aware of DNA on Loan, which is pretty much what it says. It's an older article that was written very soon after the Red Ward situation, and here is an excerpt from the abstract. The DNA is considered to be on loan to the researcher for the purpose of the research for which the consent was obtained. I would like, if we have time, to look at a 5-minute clip, assuming that I can make it work, which talks about this other point. So this is part of the research project we did, and in this situation we had focus groups with 4 different groups of Indigenous people, including one here at Langara. We did some thematic analysis. We went back to the groups. We showed them our analysis. We discussed it with them. Did they agree with it? Did they disagree with it? They gave approval or not for use of their quotes. So I'd really like to show you a little bit of that, and we can quick sort of. One second. Let's see if this is going to work. I'm hoping that there is sound. So this is part of this video.
Structure. Should there be a separate Indigenous database, or should Indigenous genomes be added to existing international databases?
So this is one of
How
the
should the
themes.
database be controlled?
Is this a database kept separate,
Just listen and stuff.
or is it with the rest of the world? And why would it be different?
I think it should be separate.
Why would somebody international be interested in our DNA?
Because people don't stay at home anymore. There might be somebody who needs the information that was.
But I would think that if somebody traveled internationally, if I traveled internationally and I got sick, say like in Spain, and they knew I had Aboriginal ancestry, they could automatically come right back to Canada, get the database information from Canada.
Well, what if you just want to, when you go to the doctor or the hospital or whatever, and you don't want to fill out the ancestry part? It's my business what I am. Treat person. So how do they know which database to look at? And then how do you distinguish if he's giving you treatment that he would give everybody else? Or is he giving you differently because you're Aboriginal or whatever?
What if a person is biracial?
Each of our family situation is different, and my children are not full.
Sorry, we can't get a picture, but that's. Now I'm going to see if I can get back to where we were with. I think you might have messed me up here. Yeah. Let's just see. Good slideshow from one slide. Yeah, now you've lost it. You know what?
I think we're going to.
Yeah, stuff happens. Let me just talk without slides because it's probably better anyway. I always come with pictures of my slides because this happens to me all the time. I wanted to talk about, sorry, this is very unhelpful. I wanted to talk about some of the REB issues that came up. Just maybe I'll just pick one because of the time constraints. This is the comment I got regarding the consent form from REB. It says, it would be helpful to include an Indigenous body. The participant could, thank you, thank you, could turn to for any complaints or concerns listed before the standard research complaint form. So what do you guys think about this? This is Children's and Women's Research Ethics Board, CBC. Is that reasonable? Well, so in my ignorance, I thought it sounded pretty reasonable. So I took it back to the team. And so there must be a way to change these slides now that my own machine...
Oh, use your computer. Use your computer to move forward. Your computer. Yeah.
Oh, that's fascinating. Okay, cool. Okay. So I took it back to the team. I'm seeing two completely different slides here. So. And the team members, the Indigenous team members, perceived it to be a really culturally insensitive request. Their reasoning was that if you're not status First Nations health authorities are going to help you. If you're not a member of the Métis Nation, they're not going to help you. There's nobody who can help you if you're an Inuk. So they found it to be culturally insensitive, and they wanted to emphasize that we're all members of different communities. I might be Métis, who's a Green Party candidate, and a student at SFU. Those are, three different groups. And they really wanted to emphasize that. I said I wanted to move briefly beyond Chapter 9. And I think that Chapter 9 was probably written before the TRC recommendations. And what the team wanted to look at was, is the project that we want to do consistent with the calls to action? And they decided that yes, it is. It's consistent with the call to action to close the gaps in healthcare inequity. And for that reason, they felt that this was a project that they could get behind. Indigenous cultural safety, are these terms that you're familiar with? The continuum from cultural awareness to cultural sensitivity to competency to safety? Everybody's nodding good, then I won't go any further about that. But to remind you that cultural safety is something that I cannot say I'm practicing. That's something that the participants have to say is culturally safe. And I would maybe suggest that REB could be maybe looking more closely at cultural safety. That phrase does not appear in Chapter 9 of the whatever it is. I'm going to say TMSC, the other thing, the ethics thing. It does not appear there, thank you. So maybe that's something that we could be looking at a little bit more. Yeah.
Sorry, I'm here because I'm not an expert in this. Can you give me an example or tell me what cultural safety is?
Okay, absolutely.
I mean, I can
Yeah. So let me just back up briefly. So cultural awareness is that, I know that your culture and my culture are different, but cool. Cultural sensitivity is that, I recognize that the differences may be important to acknowledge in research. Then cultural competency means that I'm focusing on attaining the skills so that I can respectfully integrate that knowledge into effective and respectful research. And then cultural safety means I'm considering how the social, demographic, and power dynamics are influencing and shaping people's experiences. So that's where the cultural safety is. Is that fair enough?
Yeah. So, but wouldn't that normally be an issue that would be addressed in the requirement of consultation with the relevant communities?
We don't get to talk to them until we've got a rubber stamp from you guys. Don't forget that, right?
Well, we require that you should, it's a requirement in Chapter 9 that you demonstrate that your application has had consultation with some relevance communities that are going to be potentially impacted by the research.
Yeah,
so
I agree, but I still think that and the Chapter 9 obviously is extremely well thought out and very well written, but I would just like to see cultural safety as an alternative framework for people to consider. I'm just putting it out there. I just disagree. I've had it kind of persuaded into me, and I have learned a great deal.
I think you're right, that 1, 2, and 3 are sort of uppermost in the minds of research ethics boards.
Yeah.
But we expect the representatives that are consulted to assist us, to assist the researchers, to interact with the culture in a way that is respectful and that the culture will think reflects a degree for them of their safety, right? So.
Okay. Yeah.
And just to clarify, we do allow and encourage a consultation with the community, the Indigenous community, well prior to the research commencing, because you really need to consult with the community in order to determine what the research should be anyway.
Yeah.
But there's provisions in the TCBS that do allow for those kinds of consultations, which are not considered interactions with human participants in the course of the conduct of the research.
Sure.
So you
Yes.
don't have to have the
Yes.
approval first.
Yes, yes, that's right. Good. Thank you for that clarification. Exactly. Okay. I just thought of some relevant examples of how you could look at it as a framework, and I'm sure that these are all things that we do anyway. And they're completely consistent with Chapter 9, but it is maybe a different way of looking at it. So if I'm looking at recruitment, these are some of the things that I would be looking at: building relationships, using pre-existing relationships that are available, making contacts. And I have to say that at Langara, it was, to my mind, as good as it can get. I mean, I contacted John. John put me in touch with Dr. REB, who was extremely gracious with his time, and he put me in touch. He connected me up with the student liaison officer, who's Indigenous, and so that removed the power structure from recruitment. And so that's Julie Aiken. If you know him, he did a fantastic job working with us, telling us what would work for his students, his colleagues. So that was I thought that was a really nice example. So thank you.
I suppose that what you're presenting here is on a general character, not necessarily on a genetic point of view, right?
Exactly, yes.
Okay.
I was just going to sort of whip through
There's a
these.
self-identification there.
Yeah. It's just because we do so much, we do so much genomics and genetics with everybody, but this is particularly relevant. The role of location is obvious, and again, to use the Langara example, we used maybe the space which was sort of tricky for us to use. We used the Indigenous student space, but it worked really well because it was comfortable and it was familiar, and we were going to the students rather than having them come to us. Role of elders is something that we have used intensely. Every one of our focus groups had an elder. The elder was extremely important to carrying out a project. She was able to anticipate a triggering, and I'll give you a couple of examples of that. We had several older participants who wanted to tell their story of residential school or the 60s school. And this takes time, and I had to realize how in my, kind of, as I say, Western-oriented research track, I had to realize how important it was, because they were essentially demonstrating trust in our process by telling those stories. And I had to be reminded of that. So even though it may appear to derail the focus group, actually, you can't dismiss it. It would be not only rude, but incredibly unethical to do so. I'm going to skip through ceremony, the role of food. I would like to ask you about the role of non-Indigenous researchers. How do you consider those in your REBs? Is it something that how do you evaluate them? I ask because I assumed that we were going to have a facilitator for our focus groups who was Indigenous. We were having a hard time finding one, and I said, well, there's Nancy, and she's a superb facilitator. She's got lots of cross-cultural experience and blah, blah, blah. And the Indigenous team member says, actually, sometimes that's better. The reason it could be better is because if she's affiliated with, let's say, one band, and the representative of that band there, they may be afraid to talk, or they may feel, or the other members who are not affiliated with that band may feel there's an ally situation going on there, and it can set up some difficult situations. So they felt very comfortable with that. So we discussed involvement of non-Indigenous investigators and the fact that we were trying to create allies. And they reminded us that we're working with and not for Indigenous people. That being said, we were criticized by an Indigenous academic institution because the project was not instigated by Indigenous peoples. And again, when we took that to our team and discussed it as a team, their perspective was that it was consistent with the course of action. It was addressing a gap in healthcare, and they felt that if we had this true partnership, then it would be something that was in the spirit of reconciliation, and that's why they supported the work, despite the fact that it was my idea and I mostly wrote the grant and mostly wrote the paper. That doesn't matter. I think we have to get over the fact that my part. Ethical partnership with participants is what we were going for. Somebody talked earlier about ownership, control, access, possession. What if the community doesn't want to results published, or they say, well, who owns the data? And the model we chose was a community-based participant research model because it basically addresses things up front, and it's not an us versus them, it's a we. So are we going to publish this? All of the data that we published was okayed by the participants, which is, it's a lot of work. So we went to these 4 focus groups, and then we had to go back to the same focus groups, invite them back to see to discuss our analysis and to give us permission to make a video of it. But imagine their surprise when we said, well, this is yours, I mean, you did the work. They were quite surprised by that. So it was a model that worked for us, and I think the community-based participation works really well for these sorts of situations. I think we're running out of time to talk further about genomics. Is that correct? Yeah. So I'll shut up. But what I will do is, if anybody's interested, I will send you the link to the full 25-minute video. It's a thing that I think that you and many other people have reacted to is how we used culturally appropriate, if you want to use those words, analogy to explain the issue of inequity and variability in the genome. What we did was we said, well, everybody in the world makes blankets. Everybody's blankets are different. They make out of different things. Some are woolen wool, some are cotton. Weaving techniques are different. The colors are different. And so if I compare my blanket to a blanket that's made in China, I'm going to go, oh, my blanket's wrong. But it's not. It's just fine. If I compare my blanket to somebody else's blanket, I can see, okay, somebody else in my ancestral group is fine. Then I can see that, okay, so my blanket has red here, yours is blue, but essentially the same colors, the same design is there. So we used that as an analogy, and it worked. I think it worked really, really well for people to understand genomic variability. So it's a 25-minute video. So thank you very much.
Thank you. Okay, I'm mindful of the time. We've had a very good discussion. Are there any sort of, maybe there's coffee, there's donuts. Maybe I will let's invite the discussion to continue. And yeah, thanks, Patricia. For what it's worth, I think there's a great paper and a great lesson in that should be told. I mean, it's being told partly through this video, but it might be nice to have a research paper that just tells us about what worked with this project and why it worked, and to some extent it can be a model for other people to follow.
It's been submitted.
Ah, okay. All right,
It
so,
was only a year's funding,
Yeah,
it was only $50,000, right?
so thank you again for a very illuminating talk and a starting of a conversation that I agree that I think we ought to be having. So thank you.
Thank you very much.